is Adrini, and thank you for virtually joining us for the latest version of the Variant Effect Seminar Series. And as I said, my name is Adrini, and I'm a member of the Variant Effect Seminar Series Organizing Committee, along with Ian, with Mariano, Ohana, and with the help of Alex and Lara. So this organization, these seminars are possible with the support and a lot of help from Lara and Alex through the sponsorship of the Atlas of Variant Effects Alliance. And as a reminder, we are always on the lookout for potential speakers for future seminars. And if there's anyone you'd like to see present, or if you yourself is interested in sharing some of your own work, then feel free to submit a speak nomination form. And you can also do a self-nomination, and we keep an eye on this list so we can reach out to potential speakers and make a schedule for the year. And as usual, before the presentation starts, I have some housekeeping items to mention. So each talk is around 20 to 25 minutes long, but there should be time for a few questions. Please post these questions at any time to the Zoom Q&A, and we will read them out to the speaker at the end.
All right.
If you have additional questions after the end of the seminar and you are an AVE member, you can go to our Slack channel. There is a tab called Seminar Series, and we can start a discussion there. And finally, we are posting updates on our Twitter X account, so be sure to follow us there. Additionally, you can catch the seminar today and previous presentations that have been recorded on our affiliated CMAP YouTube channel. Don't worry about trying to copy the URLs. We'll post these links to the chat once we get started. All the details of the seminars can be found in our website, including regular updates on the schedule and, of course, upcoming speakers. So with this QR code, you can find the link directly to our schedule with all the details of each one of the speakers. And for the last housekeeping item, this is rarely an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful, and we want everyone to use welcoming and inclusive language. So let's get started with presentations today. So today we have an amazing lineup of speakers. Our first talk has two presenters, Matthew Duran and Nicholas Perry. They will talk about direct programmable recombination of target and donor DNA to bridge RNAs. And the second speaker is Arjun Aditan, and he's talking about using DMS for the rabies virus glycoprotein. So with our first speakers, Matthew Duran got his PhD from Stanford University in 2020, and he was working in the Amibat and Stephen Montgomery labs on characterizing mobile genetic elements computationally. Then he did a postdoc at the Patrick Hsu lab at UC Berkeley, where he discovered and characterized new large serine recombinases for efficiently DNA cargo integration into cell human genomes. Now he's a senior scientist at the HCU lab at the Arc Institute working on programmable recombinases and genome design. Nicholas Perry is a current PhD student at the UC Berkeley, UCSF, also working at Patrick's HCU lab at the Arc Institute, focusing on the discovery and characterization of programmable recombinases. And our last speaker, as I introduced you before, Arjun Aditan, he got a PhD in bioengineer from Stanford University, working in polyphordiasis lab, where he developed high-throughput platforms for measuring transcription factor DNA binding affinities. And today he's doing a postdoc at Jessis Bloom's lab, and he will be talking about DMS for the glycoprotein of the rabies virus. So without further ado, you can go ahead, Matthew and Nicholas, and that's going to be an amazing presentation. You can share your screen now.
Thank you. Okay. All right, yeah. So today we are going to be presenting, Nick and I will be presenting together a paper that we co-led and was published last week in Nature, titled Bridge RNAs Direct Programmable Recombination of Target and Donor DNA. And we'll also briefly be discussing some work in a second bridge recombinase structure and mechanism paper that we were both authors on, which was also published last week in Nature. All right. So Nick and I co-led this project, but it was very much a team effort, and we want to make that very clear. We had several really great scientists working with us, including research technicians, biochemists, scientists at the University of Tokyo in Hiroshi Nishimasu's lab, Silvana Konerman at Stanford and ARC, and Patrick Sue at UC Berkeley and ARC. All right, so in terms of background, mobile genetic elements are extremely diverse and abundant elements that can transfer themselves between or within genomes. They have evolved many different strategies for accomplishing this, and their transposition or horizontal or vertical transfer is one of the main ways that DNA rearrangements occur, and they play a key role in genomic diversification. Many of the most valuable and useful molecular biology and genome engineering tools actually can trace their origins back to the mobile genetic elements and the arms race between mobile genetic elements and their hosts. So insertion sequences are one very broad category of mobile genetic element, and they are these minimal autonomous DNA transposons that typically only encode what is necessary for their own transposition, such as a recombinase or a transposase gene. And they usually have inverted terminal repeats at the end that are recognized by the transposase protein. And they can also uh, acquire over time additional passenger genes that can confer benefits to the hosts and ensure the propagation of the mobile genetic elements. So IS elements have been categorized by many scientists over decades to identify different families. And there's usually, or so far there have been about 28 different families that have been categorized. And they're usually categorized based on their recombination chemistry and the sequence organization of the element. And they appear widely across bacterial and archaeal genomes, and they're the ancestor of many eukaryotic mobile elements as well. Okay, most IS elements have one of four different recombination chemistries, such as DDE, HUH, motifs, or a serine or tyrosine catalytic residue. And they typically have terminal inverted repeats that are bound by the transposase protein through protein DNA interactions. And IS-110 elements are unique among the 28 different families in that they have a DEDD motif as part of their catalytic mechanism, which is actually a RUV-C-like motif. And they often don't have terminal inverted repeats. And when they do, they're usually much shorter than is typical for most IS families.
So we'll lead into how we discovered the recombination mechanism for these IS-110 elements. So IS-110 are one type of transposon that does cut and paste, in that the element, which is encoded within a genome, as part of its propagation to new hosts, will cut itself out of the genome and form a circular form. And that circular form, when it encounters a particular target site, it will paste back into the genome, either back into the same site it cut itself out of or into a new site. And we were curious as to how this system was able to identify its DNA target sites. amongst all IS elements, one of the first things that we noticed doing a computational analysis was that out of the various families, these IS-110 elements have the longest median non-coding end length. As Matt explained earlier, these IS elements are sort of defined by their minimalness. And so the fact that they would have a long non-coding end would suggest that these long coding ends serve some function. When we looked at these non-coding ends as part of the circular form of the element, when the left end and right end come together as part of the circular DNA, they form a promoter motif, which is something that other researchers have noted in the past for these IS-110 elements. What was curious about this is that the previous hypothesis was this promoter was formed in order to drive recombinase expression. However, the beginning of this promoter is more than 200 bases away from the star codon of the recombinase. So we cloned this sequence of the ends onto a plasmid in E. coli, and then we performed RNA sequencing for 6 different IS-110 elements. And what we saw was that ahead of the recombinase protein, a non-coding RNA was expressed right where we predicted the promoter to begin transcribing RNA. When we looked at the RNA sequences across hundreds of IS-110 elements based on that region, what we noted was a particular conserved secondary structure that was quite striking. This structure had these very strange internal loops. And to have this sort of lack of structure in this RNA got us thinking about what the purpose of this RNA may be. Our initial hypothesis is that, to sort of summarize, what we have observed is that these elements are cutting themselves out. And once they're cut out, this promoter forms and drives this non-coding RNA, as well as the recombinase expression.
So to try to figure out what this RNA was doing, we performed a covariation analysis of thousands of IS-110 elements with a focus specifically on IS-621 and its relatives. And using software I developed as a PhD student, we could precisely identify the boundaries of thousands of different elements using publicly available genomic and metagenomic sequence databases. And once we could identify the boundaries, we could reconstitute the original target sequence and donor sequence, and we could identify the bridge RNA using a covariance model trained on the structured RNA. And then we could do a paired alignment and analyze covarying nucleotides to identify those that might be functionally linked to each other, which could, in the case of nucleotides, indicate base pairing. And once we saw a signal, we also looked for base pairing concordance. So not only do they covary, but do they covary in a way that suggests base pairing? Then we mapped those back onto the original secondary structure to see if it made sense. And then we repeated this same analysis for the non-coding RNA and the donor sequence, so the junction that is formed after excision. All right. So yeah, this was really the first very exciting eureka moment of this project, where we saw a very, very clear signal of covariation between the non-coding RNA and the target sequence. And this implied that the two different sides of this target binding loop bound to the forward strand and reverse strands of the target, which was a really unique base pairing mechanism. And what's more, we were very excited to see that we saw a similar covarying signal for the second internal loop. But this second loop covaried with the donor sequence. And this implied a potential base pairing and programmable interaction with both the target sequence and the donor sequence. And that programmability on both ends of the mobile genetic element, both ends of the mechanism, was totally unprecedented as far as programmable recombination or transposition systems go. So this helped us to finalize a model for how the system worked, or to establish a model before we had really any experiments. We had a pretty good guess for what was going on. So it could cut itself out to form a circular intermediate, and that would form this promoter to drive the transcription of the bridge RNA and the recombinase. And then that bridge RNA could recognize both the circular donor and the target sequence by base pairing to mediate recombination between the two molecules. So here's a model for how these base pairing interactions work in this system. Here at the top, in blue, there's the target binding loop that has the two sides that bind to the forward and reverse strand of the target. And on the bottom, we have the donor binding loop, which binds to the forward and reverse strands of the donor. And over here is showing how that likely worked, just based on how we understood it so far computationally. And these base pairing interactions were very promising and suggested that there's a good chance that it's a programmable system.
Okay, so to start to try to investigate this, we just tried to reprogram target and donor sequence specificity in the E. coli system. So the way that we did this was to take the sort of important parts from the natural system, so that would be the target sequence, and encode it on one plasmid, and then sort of deconstruct the mobile element, where we put the recombinase on one plasmid and the donor sequence, which is the concatenation of these non-coding ends, onto a second plasmid. And as we observe for the natural system, recombination between this RELE sequence and the target should result in recombination of these two plasmids. And to measure recombination, the recombination between the target and the donor should result in this promoter here driving a GFP gene. So when we initially tested this, we were able to observe via flow cytometry that a significant proportion of E. coli began expressing GFP when we co-transformed these two plasmids. And to confirm that this recombination reaction was dependent on both the bridge RNA and our recombinase, we inactivated both the RUBC domain and TMP domain of the protein and saw no recombination. And then when we abrogated the donor, which encodes the bridge RNA, we also saw no recombination. To try to tackle this more succinctly in collaboration with James at RA in our lab, we tried to demonstrate that we could reprogram that assay. So we simultaneously reprogrammed either the target and the target binding loop of the bridge RNA. And what we did is we took the system and we paired the reprogrammed bridge RNA where the target binding loop specificity is for an entirely new sequence. And what we observed is that it enables robust recombination as measured by GFP signal when we supply the cognate target. But when we supply back the wild type target, we see no recombination, which suggests that the RNA DNA base pairing is the only determinant for recombination. And so we tested this concept across 7 different target sites, observing a range of efficiencies, including ones that were more efficient than the wild type sequences. Next, we performed the same thing for the donor binding loop using the same strategy where we reprogrammed the donor binding loop of the bridge RNA and simultaneously reprogrammed the donor sequence. And with a similar effect, what we observe is that we can reprogram to any donor sequence to as many as every single nucleotide changed from the original donor sequence. What we noticed as part of these experiments, which was quite interesting to us, is that even just a single nucleotide change from the wild type donor to this D1 donor, we observed almost no recombination with the wild type donor. So just a single mismatch significantly reduced the efficiency of the reaction. And so this led us to perform additional screens that Matt will lead us into.
Yeah, so this strongly suggested that it was not very mismatch tolerant. So to more thoroughly characterize this, we performed a pooled screen of thousands of different bridge RNAs and their target and donor sequences, introducing single and double mismatches at various positions to assess mismatch tolerance and overall sequence preference. And we found that in general, the programmable portions of these bridge RNAs have low mismatch tolerance, with a lot of the efficiency being completely abrogated with one mismatch, and then an even more pronounced effect with 9 mismatches. And we could also reprogram the loops. The screen showed that we could reprogram the loops to thousands of different possibilities and still detect efficient recombination. So in here, we're showing the sequence preference in the target and the donor overall sequence composition. And overall for the target, there's relatively low bias in what sequences, what nucleotides it prefers at each position. But the donor is somewhat unique in that it strongly disfavors a cytosine at position 7 of the donor. So that is one limitation of the system as we currently understand it.
So the next thing we looked into is that we are able to do recombination with these enzymes. And Cre recombinase, which a lot of people are familiar with, has been used for decades to mediate these recombination reactions. And we think of insertion sequence elements as doing an insertion reaction, and that's what Cre is actually natively performing as well. But as a genome engineering tool, it's also been used to excise sequences and also invert sequences. And all of this is dependent on the relative positioning of the LOXP recombination sites, whether they're on two different molecules, on the same strand of the same molecule, or on opposite strands of the same molecule. So we thought that we could recapitulate these three modes that are used for non-programmable Cre with a programmable IS621 in E. coli. So the way that we set this up is to first perform a transformation into E. coli of the bridge RNA and the recombinase to drive an insertion of a donor sequence into the E. coli genome. And we analyze our insertions using whole genome sequencing. What we observed is that when we reprogrammed bridge RNAs to target new sequences in E. coli, we got reasonable specificity ranging from about 50 to 90%. What we noticed, however, was that the right target guide, one of the two sides of the target binding loop, only encoded four base pairs. But our computational analysis suggested that 7 base pairs would be possible between this right target guide and right side of the target. When we increased the specificity of the right target guide to base pair with 7 bases instead of 4 bases, we saw dramatic increases in both the specificity of the reaction, and then we also saw fewer off-target sequences when performing insertions into the E. coli genome. Next, we designed plasmid reporter systems to try to recapitulate both programmable excision and programmable inversion of DNA sequences in E. coli, demonstrating robust recombination using both modes of rearrangement in an E. coli system. And so we think that this demonstrates that the IS621 and IS110s in general are a promising mechanism for doing programmable versions of many recombination reactions that we're familiar with using Cre.
Great. Thanks, Nick. Okay, so here we want to, that really kind of summarizes the first paper, and we want to start briefly describing the structure and mechanism paper. This was done in collaboration with Hiroshi Nishimatsu and Masahiro Hiraizumi at the University of Tokyo. They were able to resolve the full recombination complex with cryo-EM. And here's the video of that complex. So in yellow, so actually each protein dimerizes with another protein in its coiled domain. and that binds to the non-coding RNA, which directs it to the donor and the target. Here you can see the donor RNA and the donor sequence base pairing as expected based on our computational analysis. Yeah, there's the dimer, the protein dimer binding. Blue is the RUV-C domain. Yellow is the TNP C-terminal domain. Here's the target RNA, target loop binding to the target sequence in a similar way to the donor loop. And yeah, overall, the RNA sits between the dimer and the DNA. Here's the full kind of tetramer of nucleotide sequences. You can see they come in very close proximity with each other. Here's just the tetramer of the proteins. And yeah, the full complex. So that's brief overview of what's going on in the structure, and it largely confirms what we saw with our computational predictions. And one really cool feature of this system, which is actually kind of a similar theme along with many other recombination or transposition systems, is that these kind of transverse protein dimers, they only can catalyze recombination when they come together, and the serine loop in the C-terminal domain can cross across that interface and interact with the DEDD motif in the RUV-C domain, coordinated with a magnesium ion, form a 5' phosphoserine covalent bond that then frees up the top strands to move across the recombination junction to form a holiday junction.
So as Matt just mentioned, what our structural work was able to uncover is 3 different states of the reaction. So what we looked at in the video is a structure showing the complex bound to DNA and RNA. So this is the DNA isolated from that structure. And what we observe is that the top strands of both the target DNA and the donor DNA excuse me, have been cleaved via a covalent phosphoserine bond. But we were also able to show structures where, after the cleavage occurred, strand exchange and religation of the top strands has occurred. And so this results in a holiday junction, which is a feature of many other DNA recombination mechanisms, both sort of unintentionally during DNA repair processes, and then also with other recombinases or transposases. And then finally, we were able to show that this holiday junction is resolved via cleavage of the bottom strand. One interesting thing about our structural work in sort of concert with our computational and experimental work is the emergence of actually an additional component to understanding how to work with bridge RNAs, which we've called handshake guides. And what these are is, as we showed earlier, the target binding loop binds the target, and the donor binding loop binds the donor. But encoded within the target binding loop are these handshake guides, and these bases encode specificity for the donor. And these bases on the donor binding loop encode specificity for the target. And what we think is happening here is that these new interactions promote the strand exchange reaction that we just mentioned in order to drive this reaction forward. So I encourage you guys to read the structure paper if you're interested in this mechanism further, and feel free to ask questions at the end of the talk.
All right, so what is next for these bridge recombinase systems? So of course, we're very interested in exploring the diversity of these systems. Here we focused on IS621 and related orthologs experimentally, but we've computationally characterized many, many more sequences than that, and we want to understand their mechanisms. And one important feature, that defines the family overall, is that IS-110 group, which is one of 2 groups that fall under the IS-110 family. IS-110 group elements typically have their bridge RNA in the 5' LE sequence, and we've been able to detect target and donor binding loops across these diverse sequences. Whereas the IS-1111 group, which is the second group in the family, they tend to encode their bridge RNAs in the RE sequence, and we also can detect target binding loops and donor binding loops for that group. So really we've mostly only focused so far on the insertion mechanism, but that's really only half the story. So we're definitely interested in understanding excision in more detail. And we're really excited about this because we think it could represent really a third generation or a new generation of programmable RNA-guided systems, beginning with RNAi and then CRISPR. We think that bridge recombination really could open up a wide variety of new structural variant rearrangement technologies that we're very, very excited about. So yeah, beyond CRISPR, with this system, we can start thinking about much larger scale chromosomal and genomic variants. And we really could use this system to engineer or to study or to create these new variants. And of course, we could potentially use it to reverse deleterious variants and restore a healthy function to the genome.
So just to summarize, IS-110 elements encode a bridge RNA to mediate recombination. Reprogramming this bridge RNA enables insertion, excision, and inversion of almost any two sequences of DNA in a reprogrammable manner. And that these systems are promising as a third generation tools. And we think we can enable design of whole genomes using these tools. And then finally, further study of their recombinase diversity, the mechanism, and the structure will enable us to have a robust understanding of these enzymes so that we can apply them towards all of these promising applications. So with that, we'll thank all of our collaborators as well as our lab. And finally, we will take any questions. One thing I'd like to mention before we end is that we have resources for applying this in your own lab. So both classes on AdGene, some code for how we've analyzed our data for our paper, and then also a tool that will help you design bridge RNAs for your application.
Okay, thank you very much, Matt and Nicholas, for this amazing talk. I have two questions mainly. The first one is regarding the structure, this secondary structure of the loops. Besides the base pairing, how important is this structure of the loop?
Yeah, I can help answer that. So and why don't I flip back to the very beginning, that just shows the conserved secondary structure. So this is very similar to what we observe in our structural determination. Obviously, it is more twisted, so to speak, but the loops are open in the center. As far as critical residues, you can see here that there's a sort of bulge of a single A on both sides of the loop. And that's actually found on both the target binding loop and the donor binding loop. It's a little bit more vague, I guess, in the secondary structure. But what we notice is that the recombinase principally interacts with these A bulges in order to bind to the RNA. So I'd say that's the main secondary structure implication.
Okay, thank you. And then what I was thinking is if you have tried to edit some mammalian cells, human cells with this system.
Yeah, this is something that we're still working on, and we're not ready to share data or
Okay.
information.
And then I have a question from Ohana. She's asking, how do you filter to explore IS-110 recombinase among the others?
Yeah, that's a good question. So I would say there's actually, it stood out in that we really didn't understand the mechanism, and there were some guesses, but there was very little experimental evidence to demonstrate the mechanism. People had tried to reconstitute recombination with these systems in vitro, couldn't get it working, which implied to us they were missing something. And then yeah, I've spent my, since my PhD focused on characterizing these mobile genetic elements, understanding them, and this one kind of always stood out to me as an interesting one that we could go after. And when I started looking at examples of the element, I just noticed, okay, this has an unusually long non-coding end. Usually they're much more compact than this. So maybe there's something going on there. We don't know how they work. They have these long non-coding ends. They also have a REV-C domain, which we know associates with RNA guides in the form of CRISPR nucleases. So all those things together kind of narrowed us down on this as an exciting potential RNA-guided recombinase.
Okay, thank you again. There's a question in the chat, but it's